Good morning and welcome to the Nature Research Center. Thank you for joining us in the Daily Planet. If you didn't see our presentation yesterday, you'll learn that you are here in a very special week. This is our third year of a grant project that's funded by the National Science Foundation. And in this grant, it's called Students Discover. And we bring in middle school teachers, 12 teachers each summer, to work with postdocs. We have 5 postdoctoral scientists leading 4 projects, and 3 middle school teachers work with each, and they co-create the science and lesson plans that allow the teachers then to go back into their classroom and engage the students in real scientific discovery. And so these are not messing around scientific discoveries that get published in the scientific literature because the scientists are looking for academic jobs, but it's also really exciting, and as our teachers come here as teachers, they realize that they leave as scientists. And so yesterday we had our first team, Team Soil, that was working up in the genomics lab, telling us about their project and their experience here. And today we have Team Ant, and so I'll let them introduce themselves and come up, and we can hear about Team Ant and Ant Health Watch. Let's give a big hand.
All right, hi, I am Paige Derwin. I teach 7th and 8th grade science at Wake Young Men's Leadership Academy. Thanks to my people for coming out.
Hello everyone, I'm Maggie McKinley. I teach 7th grade science at Burgaw Middle School in Pender County.
And I'm Michelle Hafey. I teach 7th and 8th grade science also in Pender County at Penderley School. So we've been spending the last couple of weeks working on projects with ants. So why ants and why now?
So one of the answers to this question is that ants and insects in general are incredibly diverse, much more diverse than a lot of people realize that they are because they're so small. So this graphic is showing the number of different species of both mammals and insects. Mammals get a lot more attention, which is why their picture is so giant, because they're big and they're furry and they're cute. But actually there are way more species of insects, so the picture really should look more like this. We're not biased at all.
So our whole goal is to engage students in being able to discover this diversity. And so we've spent the last couple of weeks doing lesson plans so that teachers can implement these projects within their classrooms and compare them to the already existing online data in CODAP.
So the first project that we worked on when we got here is called Ant Picnic. It is a global citizen science project, which means that people all over the world, teachers and students and just regular citizens, are able to participate. And what they're doing is they're setting out different baits for ants so that they can see what ants prefer in different environments, whether they're rural or in a cold place or a warm place. They can see how that compares to other places.
So here we have our start to the project. I know it looks like we're just in the car riding around, but this is urban science, and this is how we got from our sites. So we had to drive a lot. It was about 8 hours in the car, and that was a long day. But we made our baits for the ant picnic. We made that the night before, which is, we had cookie, we had sugar, salt, amino acid, olive oil, and water that we had already prepped and ready to go, cotton balls sitting in mason jars. So once we got out to our sites, we could jump out. We set out our note cards, and then we had our cotton balls on each note card and a cookie tray to hold the cookie that we crumbled. And then we would try to stake them down with these little metal stakes because it was very windy, and if we did not hold them down, they would have been gone. Although we did have a couple culprits steal some things, blaming the squirrel and the cats. But we did have a lot of ants at some of the sites, so we had to loop back around. It was 19 sites that we drove to in Raleigh, and then we had to go back around to the same 19 sites and pick it up, which was a little challenging because the stakes did not come out of the ground very easy, so we did have to try to get the ants really quickly, get them in a Ziploc bag. Took a little bit of teamwork there, and if we lost the ants, we have our Antspirator necklaces that we were trying to catch them really quickly as they started to escape.
So the existing ant picnic data is on CodeApp, and with this online analysis platform, students can access the data. They can click on a country. It highlights all the information for those sites in that country. But there are other ways that students can also access the information. If they scroll down to the bottom, there is a world map, so when they select their location, when it's selected, you can see biome information about that site, and then scroll back up, and once again, the data table's highlighted. The beauty of this tool for students is that they can actually start analyzing some of their own ideas and questions using the existing data, and so making graphs makes it easy for them to come up with whatever their claim is, and then provide the evidence to either prove it or disprove it.
So when we did Ant Picnic, we collected thousands of ants. We're still not done counting them. I counted 572 ants from one cookie bait, which is a lot. But the question is, now that we have all of these ants, how can we identify them? How do we know what they are? So, question for you all. How many different ant species do you think live in Raleigh? Yell it out. 10. So when I started this project, I really didn't know anything about ants, and I pretty much thought that there were really 2 or maybe 3 types of ants around this area, which are the small ones that are in your kitchen, and they're pretty friendly if you don't bother them, and then the really mean ones that are pretty aggressive and really not friendly at all. But what I've learned in my last few weeks here is that in reality there are so many species of ants that are living here in our backyards. During our time doing Ant Picnic, we found 16, and there are way more than that we didn't find. So up here you can see some of them. Our postdoc Magda took these pictures, which is pretty amazing. So now that we have them here, the question is, how do we use these pictures and their body types to tell what they are?
Now, as we start talking about taking our students outside to actually do some of these labs and actually going out there to collect ants, so as a teacher or as a parent or as a participant in the activity itself, what might be your biggest concern if you're out there actually having to pick up ants? Which one would be your biggest fear? Getting bit by which one? Fire ant. Okay, nobody wants to come across the fire ant. They are usually the ones that people will jump to and they're like, oh, it's a fire ant. Well, a lot of them are not fire ants, but a lot of them are fire ants, and I personally did not want to touch the fire ant. But the question is, how can you tell which one is a fire ant? So here are some pictures of ants. They look relatively similar. Do you have a guess of which one is the fire ant, A, B, C, or D? We have C. C? Well, the winner is B. This is our fire ant, and there are a few ways that you can tell, and this was one of the biggest things that I was concerned about. If I'm taking my students outside, I want to know which one is the fire ant because there is a lot of danger there if students have allergies and things like that. So there are some ways you can tell. One of the first ways is obviously the color. It is a reddish color, but if you look towards the back of the abdomen, which is called the gaster, it is darker in color. And they are shiny. So if you get down, you look closely, they will have a nice shine to it. And it's pretty obvious when you look at it. Once you know what you're looking for, you're like, oh yeah, it's shiny. And then they have what's called a petiole. They have 2 nodes on their petiole. So they have a petiole and a post-petiole, which hopefully you can see in that picture. And it looks like there's a big separation between the abdomen and the thorax. So it looks like it's just a skinny little line separating it. When you get close, you can see it. I didn't think I would, but I'm like, yep, I see it now. Another really cool thing is when you look at a big cluster of ants, usually they are all relatively the same size, but fire ants are all very different in sizes, and it's pretty noticeable when you look down at a big cluster. You have some really big ones, you have some really small ones, some in the middle. It's pretty obvious, but they all are different sizes. And then, most importantly, if you are standing on their ant nest, you are going to know, because they do have a pretty nasty sting, as you can see in this picture, the stinger. Now they can bite as well. I was picking up the fire ants on the first cookie bait, and one did bite me. Not so bad. I kind of flicked it off before it did any more damage, but the sting is pretty rough, so we want to stay away from that. Yeah, now you know.
So with all the ant diversity, a lot of these features are not clearly seen with just the naked eye. So one of the things that we were able to do is to mount ants. And by mounting ants, we had the opportunity to see what these features looked like under a microscope, so that we can make those determinations as far as what's familiar for certain species over another. And I didn't realize that there were so many different protocols associated with ant mounting and just the whole process. It's very tedious, much worthwhile, and then the fact that they all have to be facing to the left.
All right, so our final project that we worked on here at the museum was called Invisible Pathogens, and it is focused on fungal pathogens and how they're affecting ant populations. So right now, we are not sure what types of pathogens are affecting ants. We do know that ants don't get sick as often as humans do, and one of the questions surrounding this project is, why is that? So this is a fairly new project. We are really excited to be a part of it because the global data that exists for Ant Picnic doesn't exist yet for this project, which is awesome. And then this picture is really cool. This is also one of Magda's pictures. This is called a zombie ant. So what has happened is there is a fungus that has infected the ant, and actually it's growing now out of the ant's head. So the ant is dead. And the fungus has turned it into a zombie.
Our procedures for the invisible pathogens, first we have our aspirators or whatever collection tool, and we had to go out and find the ants. We wanted to make sure we cleaned our tools really well because we didn't want anything contaminating the samples we had, so anything we grew would be specifically from the ant. So we would collect a bunch of our ants and we brought them back into the lab, and we had petri dishes with an agar on it. It is called a rose bengal agar, which is designed to promote fungal growth and to try to keep bacteria from growing. So we actually had specifically what we wanted to grow. So we would very carefully place our ants onto our plates, which is tricky because once you get one on there, they are fast and they will escape, especially which one? The Formic ant is very, very flighty. But we would put about 3 on each plate if it was small. If you had a very large ant, 1 is probably plenty. And then we would time it, leave them on there for about 30 minutes to walk around. And what they do is they kind of clean themselves off. So they would rub their antennae, and anything that was on them would kind of get rubbed onto the gel. And then after 30 minutes, we would very carefully take the ants back off, and then we would seal our plates shut, and we would put them into an incubator. Now, we had a very fancy incubator, which is easily accessible to everybody. We were using Tupperware because some of the equipment is expensive, so we want to try to make this as cost-effective for teachers as possible, and we can get Tupperware. That's easy enough. So we would incubate it, and then about 3 days, probably about 3 days, you would see some growth.
And so what you're seeing here is a picture of the first time that we did the experiment, just to kind of get an idea of what the procedure was like. And you can tell that all 3 of our petri dishes have slightly different fungi that's growing, and they were all different ants. And so the diversity of ants is also going to lead to a diversity of maybe the fungi that may be present in certain areas. And then these were the plates that we did with our last set of ants. And so that was before they had a chance to even start growing. The ants had just come off the plates. And so what we're hoping to do is the 3 of us would like to pilot this lesson within our classroom so that we can get everything worked out and then eventually pass it on to teachers and students within the state so that they can also work on the invisible pathogens and test the ants in their area for the different fungi species.
So I think I speak for all of Team Ant when I say that we have had a great learning experience here at the museum. I have learned so many things about ants that I didn't know anything about before, so it's been pretty awesome, and it will be really exciting to share that with my students this year.
And we are now the crazy people walking down the sidewalk going, oh, there's ants. And we will stop to try to identify them. And I can proudly say that I can identify quite a few now, which I never thought would be something in my life.
And it is kind of infectious. Last night, while in my hotel room, I got the black pipe cleaners, and I actually had to make an ant of my own, and found out that it is morphologically correct. So I've learned a lot, and I really want to take this back to my students.
This is Dee Dee.
Yeah, Dee Dee.
And one last thing about this ant. This ant is called the crazy ant. And I learned about this ant because when we were driving in the car during our long time together, I said, I really hope we're going to find some crazy ants. And I was just saying interesting ants. But Magda said, well, we're probably not going to find any crazy ants today. And these are actually called crazy ants. So you learn something new every day. Now we're going to roll. Thank you.
Thank you, Team Ant. Great. So now, anyone who has any questions for Team Ant, happy to take them. How does the aspirator work?
Ms. Page, can you
Yes.
demonstrate how the little ant aspirator works?
Certainly, I would love to do that for you. So the first time that I was asked to use an aspirator, I was extremely nervous and I caught one ant, but I have greatly improved, so I can definitely show you. This is where the ant will get sucked up, and actually inside is a netting, so that prevents the ant from going down the tube and into your mouth, which was my initial fear. So you just put it in your mouth and then you run around chasing ants until you get some in your vial.
It has a filter. The filter is a pretty nice thing. Some of them don't have the filter, which this catches a lot of the dirt, so you don't get that in your mouth. Without the filter, you might get a little mouthful.
So when I was catching ants to do invisible pathogens, I was catching Formica, which is a pretty big black ant that you definitely have seen before. And they actually emit formic acid when they're stressed, and they're definitely stressed when they're inside this vial. And so you can actually kind of taste the formic acid when you are trying to catch more ants, which is a little unpleasant, but it's fine. Any other questions? I have not had any ant dreams, but I'm sure they're coming.
What do you think is going to be the most exciting part of this project when you take it back to your kids in the classroom? What will they like best?
Well, I think my students are going to be a lot like Paige said that she was, as far as I don't think they're going to recognize that there are that many different types of ants. And so I think that that's going to be opening up a new world to them.
I think my students are going to like getting outside and actually being out there doing work and having stuff that we can easily bring back into the classroom. And especially once we start using auger plates, the fungal growth happens pretty quickly, which they're going to like to see. And it gets pretty exciting. We were really excited, and we were like, oh, we have stuff growing. And I think my students will really, really like that. And once it starts growing, it takes off. So they'll see some pretty quick growth there.
I would agree with what Michelle and Maggie had said. I think my students will be really excited to be participating in real science experiments and getting to go outside and do ant picnic and do invisible pathogens and do these real experiments that real scientists are doing.
They're everywhere.
So how many species of ants are there in Raleigh, and what are you all drinking all the time?
I'm not sure about the Raleigh question, so I will defer to my scientists.
So we actually don't know how many species of ants there are in Raleigh, but in North Carolina there's about 180 species.
Is there any research on the strength of ants? Because I noticed sometimes I see pictures of small ants carrying big crumbs, and the weight of an ant versus the weight of the crumb, and what ratio that is in terms of strength. And there's a song about moving the rubber tree plant and all that stuff. So if you could help us with that.
So I don't know the exact ratio. I'm very new to ant science. But I do know that they are very strong. And when we went to do our ant picnic, we actually had some sites where ants had taken away a whole cookie, which was pretty amazing, fire ants. And then actually one day we were coming back from dinner and we saw these ants that were capturing larvae from other ants, and they were gonna make them work for them as their servants. Deanna has something to say.
Thank you. So to answer your question, what we do know about ants and how much they can carry about 100 times their body weight. So they definitely saw that out in the field. So they were carrying away cookie crumbs, but we see them carry away, larger insects.
There's a lot of teamwork with the ants as well, so you'll see a few of them working together to carry an object also, so they could take something even larger than just what they could carry themselves.
So why are all those ants women? Female? Is it because we're stronger?
That's right, that's right. I know we talked about it.
So the male ants actually all have wings. And so if you see an ant that's just crawling on the ground, you can assume that it's a female. The male ants, I believe, are only used for mating. And the queen ant is the only ant that mates, that is a female. So all the other ones are just working ladies.
Do you have any other questions? Anybody on the second floor? Yes. Our new Daily Planet curator here.
Hi, thank you for the introduction, I guess. So what were some of the results of the ant picnic? I'm thinking, was there like a surprising result? Like they really like protein or amino acids and they didn't all just go for the cookie every time? Because I would go for the cookie, but.
We haven't had an opportunity to count all of the ants from our for our data. However, we did find in some areas the cookie was the most popular, and I believe it was 572 that we had at one of our sites. Another thing that we were looking at was the temperature of the soil near the baits at the 19 different locations. So we used an iButton that would measure that temperature, and that hopefully when we start analyzing our data might give us some more insights, because there were some areas that the olive oil was actually much more preferred. So we're thinking that some of the conditions that the ants are living in may determine what it is they're seeking out as far as their food, what they may be lacking. So, but that's something that is interesting for us to look at. And with the online data, in the future, our students are going to be able to enter their data from Ant Picnic into CodeApp as well, so that they can make those same comparisons and look at those biomes that are similar in different areas all over the world.
So when I see ants in our kitchen, I just want to exterminate them. And I'm wondering if you could just touch upon any of the positive aspects that ants do for the environment or our ecosystem, so that we don't feel like we need to exterminate them all.
I'm not sure of the answer to that.
So typically when we think about ants, most of the ants that you see in your urban environment make up a small portion of the overall diversity of ants worldwide. And there are a number of ants likely that we haven't even identified or discovered yet. But what we do know that ants play a pivotal role as what some people call ecosystem engineers. They do a lot of soil turnover with their colony making, so that helps plant production. They are very involved in various interactions, so involved in scavenging and decomposition, and also supporting other species that are important for ecosystem function. So, and overall, they just contribute a lot to the diversity, which we know that more diversity usually means a healthy ecosystem. So, I would say next time you see an ant in your kitchen, just know that it's a small percentage of what's out there, and that should be hopefully exciting for you. Yeah. There's more out there.
And when you see an ant in your kitchen, take it, crush it, and smell it.
And it smells like coconut, and it's tapenoma.
No, it smells like blue cheese.
Or coconut.
Well, and I mean, I think the other really cool aspect of the invisible pathogens, like what you guys are doing is super cool, because essentially by looking at the fungal pathogens, particularly in response to different heat habitats, like cooler and hotter places, they might serve as essentially canaries in a coal mine in terms of being, that's the whole point of that, to see if they're more sensitive to pathogens that are predictive of other ecosystem imbalances.
What creatures locally eat ants?
Yeah, so lizards, let's see, other insects, frogs, spiders, yeah, other ants, which you saw, which our team saw this week, so. And birds.
So I won't ask a question about ants. So since this is a citizen science project, you're middle school teachers, what kind of support do you think that you need for the classroom or in your schools and in your districts to be able to implement these sorts of ideas, these sorts of projects moving forward, and make them sustainable? Because they sound really exciting, and I can see if, 7th grade me would love to be outside doing something like this, as well as 8th grade me and 9th grade me. So what do you need to make sure that you can keep doing these sorts of cool things with your students?
I think for me and at my school, administrative support is one of the first things, because as soon as you say you want to take students outside and you're going to be messing with ants, depending on your administrator and the trust with you of, like, can you keep your kids safe, that might be an issue, because if they don't really understand what you're doing, they may not be as supportive. Whereas some might be like, oh yeah, do it, get your kids outside, and they'll love it. Another thing is some of the stuff is expensive. So especially the rose Bengal auger, it's not cheap. So that's gonna be a little bit of a struggle, because you would want enough plates for all of your students. So having the funds to make sure you can do that is definitely important. Also, we would want some other equipment. Like in my school, we don't have a lab at all. We have one microscope that works correctly, and we don't have a dissection microscope, which we would want if we eventually want the kids to be able to look at the fungal growth up close, and to look at maybe identifying ant species, things like that. So having the materials, or at least somewhere that we can get them from or borrow them, would be extremely important.
I think another thing is making sure that your administrators, the people that are supporting you in this, know that you're tying it to curriculum. And so it does tie in with some of the science standards, but with the addition of using CODAP when they analyze their data, it also brings in a lot of math and technology standards. And I think just making everyone around you aware that this is something that is going to be a learning experience for your students. For me, I really feel like my students are better at being learners when they've had experiences like this, because it gets them excited about learning.
And to point out, though, also along those lines, this grant is called Students Discover, and we have many partners involved in it. And one of the partners, particularly that's important in terms of this aspect of scaling, is the Science House. And so they have at the Science House here in Raleigh and in satellite offices, they're essentially like the education extension offices where you can go and check out equipment. And so this is our third cohort of teachers, and this is really the beginning of their intensive year-long journey with us. And so if you look at studentsdiscover.org, you'll see some projects from other teachers that are already in those more further stages of implementation.
Do ants go back to prehistoric times? Or maybe how far do they go back that we know of?
So ants, the origin of the first ant was about 150 million years ago, and they radiated 100 million years ago with angiosperms. Angiosperms, flowering plants. Yeah.
Magda can answer any question you have ever had about ants.
Well, thank you all so much. Let's give a big round of applause again for Team Ant. Thank you so much.